Welcome back, friends. We are continuing with our topic of coordination, and in this topic we are going to discuss about cytokinins. Now, cytokinins they have somehow the different effect as compared with gibberellins and auxin, but the effect also depend on the presence of auxin. So cytokinin, they are a group of chemicals that influence cell division and shoot formation. Cell division and shoot formation. One among the very important concept for you to know here is that they promote cell division. Very important concept for you to know. They promote cell division. It is very, very, very important for you to know. So, in discovery of auxin, what the fact behind, or what they did until they discovered auxin? Until they discovered cytokinins. So cytokinins, they were called as kinins in the past, when the first cytokinins were isolated from yeast cell. Kunaweza kuona kwamba the first cytokinin were isolated from yeast cell, and they were known as kinins. So what made them to be known as kinins? The term kinin was used for substances concerned with control of cell division. Later, the term cytokinin was adopted, partly from cytokinesis. What is cytokinesis? Meaning cell division. So cytokinesis is cell division, and because cytokinins, having the name kinins, they stimulate cell division. Now their name changed from kinins to cytokinins, and partly because kinin have entirely different meaning in zoology, that it means it is the bloody polypeptide. So in studying animals, that is zoology, we have kinin, and kinin is one among the bloody polypeptides, so it is one among the protein found in the blood. So kama tungeendelea kuita hii phytohormone kama kinin, ingesababisha wanasayansi washinde kuelewana vizuri kwamba kinin which is which, kwamba kinin the blood polypeptide or the plant phytohormone. Kwa sababu waliona kwamba hii hormone inafanya kazi ya kustimulate cytokinesis, which is cell division, wakaamua ipe jina cytokinins. In terms of their synthesis and distribution, cytokinins are most abundant where rapid cell division is occurring. Rapid cell division, very important to remember, particularly in fruits and seeds, where they are associated with embryo growth. So in mature plants, they are frequently made in roots and move to the shoots in the transplantation stem, that is in xylem. So as you can see, even the route of transmission, or the route of translocation of these hormones in plants is different from that of auxin. Although they move through phloem, but cytokinins, they move through xylem from roots to the shoot by transplantation stem. Now, as you can see, this is kinetic, and as you can see, the kinin had the skeleton consisting of the cyclohexene and then the cyclopentene. Cyclohexene because it is the cyclic compound consisting of 6 carbons. 1, 2, 3, 4, 5, 6. Actually, it is not 6 carbons, but it is the cyclic compound, and it contains 6, I can say as 6 angles, because here we have nitrogen and here is nitrogen. So we can't say actually this is a cyclohexene, because it is made up of not all the not all the atoms they are carbon. Here we have carbon, you have carbon, you have carbon, you have carbon, but here is nitrogen and here is nitrogen. So 2 carbon in the cyclohexane, they are replaced in the cyclohexene, they are replaced with the nitrogen, and its name is 6-full adenine synthetic cytokinin. This is produced industrially, and it has the attached group here. Also we have the zeatin, a neutral cytokinin. Also, as you can see here, the basic structure or the basic skeleton is present here. Also we have the attached group. Again here there are another type of cytokinin, adenine, related to cytokinin. So this is not cytokinin as such, but it is the adenine, as we shall study later in genetics about the nucleic acid and the organic bases. So in loose, cytokinins, first they promote cell division in apical meristem and cambium in the presence of auxin. Very important to remember, in the presence of auxin. That means in the absence of auxins, cytokinins, they can't exert their effect, but in presence of auxin they can do their effect. The second thing, they promote lateral bud growth. This effect is opposite from that of auxins, because auxins, they tend to inhibit plant growth, but the cytokinins, the auxin, they inhibit lateral bud growth, but the cytokinins, they promote lateral body growth. Also they break down seed dormants and bud dormants in the presence of light and a suitable temperature. So tukiwa na light na suitable temperature, cytokinins, they break seed dormants pamoja na bud dormants. Then they delay leaf senescence, yani jani kuzeeka, leaf senescence they delay. Kwa tukispread cytokinins, tafanya majani yasizeeke kwa haraka. They promote stomata opening, that is clear, no need of explanation. Then they promote fruit growth and can rarely induce parthenocarp. Kwa hasasa ni fruit growth kwa sababu ya kustimulate cell division, lakini rarely they can induce parthenocarp. Sometimes they inhibit cell expansion. Sometimes. Kwa nini? Kwa sababu unapotaka kustimulate division ya cell, yani cells ziweze kudivide faster, lazima usababishe zile cells zisi expand sana, kwa sababu ziki expand sana, their rate of division will be decreased. So the faster division of cell can be achieved by inhibiting the expansion of cells. In terms of commercial application of here it is cytokinins, it is not auxin, it is cytokinins. So in terms of commercial application of cytokinins, they have about 3 commercial applications. They have about 3 commercial application, and the first is that they used it to prolong life of fresh leaf crops. Kwa wale mazao ambayo yanakuwa yana majani, na majani ndio kuishi kwa hiyo mimea, unakuwa na prisia cytokinins ili ziweze kuprolong their life, ziweze kuishi muda mrefu. For example, ni cabbage na lettuce. So they delay senescence, kama tulivyo discuss kule kwenye function. By delaying senescence, yani kuzeeka kwa majani au kuputika kwa majani, basi zinasababisha life iwe more prolonged. And then they are used to keep flowers fresh. Kwa maua unaweza keep yawe fresh yasinyauke by spraying them with the cytokinins. And then they are used to break dormants of some seeds. They are used to break dormants of some seeds. So this marks the end of our discussion on cytokinins. In the next session we'll be discussing about the ethene or ethylene, very simple organic compounds. We discuss all the facts about the ethene, and that will be to almost near our discussion of coordination will be almost ending in next few sessions. So don't forget to subscribe this channel and to share the link of this video to your fellow students. Thank you everybody. For the need of notes, you may contact me with the contacts which are present in the description below. Thank you. Let I wish you nice studies.